Okay, now we get to the fun part. So we're going to have question and answer session. We have expert panelists who are going to join us up front here. All of you know who you are. If you can come up, and then we'll tell people who you are, and join us on the panel. Perhaps while they're doing that, we have a special guest, Dr. Shakun Malik from the National Cancer Institute. She's our lung cancer representative there at CTAP, recently of the FDA, and she has worked with us for 18 months to get this protocol to its launch. And I might just ask Shakun if you have any comments, or do you want to say anything? Okay, thank you so much. All right, so I'm going to introduce our panelists, and Phil Mack. Phil, you're over here. So going down the panel, first is Phil Mack, a co-chair of Translational Medicine for the SWOG Lung Committee. Next, Shannon McDonough, biostatistician who will be working with us on the program. Next to her, Jim Moon, who most of you know, also biostatistician for the Lung Committee. Next, a recent addition, I think, to SWOG, Crystal Miwa, protocol coordinator. And then our Lung Committee coordinator, Jennifer Scott. Maybe you guys could just, you're supposed to be standing, but I guess you could just wave your hand. Dana Sparks, who everybody knows as the Director of Operations and Protocols. Nathan Erickson, Chief of Administration. Richard Cecil, the Grants and Contracts coordinator, somewhere coming up. You know, we have some shy individuals here. Okay, and then Frank DeSanto, Communication Public Relations Manager, Matt Boren from the NCI Pharmaceutical Management Branch for Drug Distribution. Thank you for joining us. And Travis Quigley, Director of Operations at Foundation Medicine. Okay, so I think I've got the lineup. Okay, so of course we're going to have our presenters stay here. We will entertain questions. You can come to the microphone, tell us who you are, ask your question, or if you're the shy type, you can fill out these little pink forms. We already have some questions that have been submitted, and so I'm going to start with those until those of you want to come ask us some questions. So the first of these I'll handle myself, and the first question is, what is the benefit of pre-screening? Because some of you may not be familiar with that term and what we're trying to do here. So the goal is to expedite getting patients onto this master protocol right after they have progressive disease, because many patients will be symptomatic, and not to delay them entering into the protocol. So if you have a patient with squamous cell lung cancer and they are getting first-line platinum chemotherapy, this allows you to sign them up for pre-screening, submit their specimen, get the result. Well, not you don't get the result, we get the result. But then when that patient develops progressive disease, assuming they're otherwise eligible, they're ready to go. In other words, we already have their molecular profiling, so you save that 2 weeks for your patient, you know, of us sending off the tissue and getting the result back. So my own estimate is that this will increase our accrual by approximately 25%. I know personally my patients would like to have that done, and they're going to want to get this done so that they can go on to the trial as soon as they have progressive disease. Any other comments on the pre-screening, Valley? Any?
No, I think you have covered it, David. The hope is we'll put metrics into the protocol to look at how successful this is going to be. We know it's going to be successful at some sites, but we would like to encourage everyone to think about it because it will expedite the patient's entry.
Great, good comment on that too. So we are going to look at the metrics and see how these people who are pre-screened do and compared to the patients that are screened at the time of progression to make sure that it's an efficient mechanism. Okay, first question, Suzanne.
Suzanne Arnold, University of Kentucky. Question, at the end of the study participation in whichever arm sub-study you get to, will you consider or are you going to share the Foundation Medicine results with the physician that's treating the patient.
Okay, and maybe what I'm going to do is if somebody wants to tackle any question, the panel, the presenters, raise your hand so that we don't have 15 people going to the microphone, because I know everybody knows the answer to this. All right,
So-
anybody, Valley?
Yeah,
Okay.
I think I should make that clear. So the results, the full panel is going to be shared at the physician's request and the patient's request at progression with the physician and the patient. It will also be shared when the study closes. So if a study does not go from phase 2 to phase 3, or even at the end of phase 3, but I think the timely thing for our patients and the clinical reality is progression, which is the crucial thing.
And if I can just amplify on that a bit, I think everybody in this room knows that we're talking about squamous cell lung cancer second-line therapy. In other words, we're not talking about EGFR mutated cancers frontline or ALK-positive cancers, which are almost non-existent in squamous cell lung cancer. If the patient does have that rare squamous cell lung cancer with those abnormalities, you will be notified up front. You probably know the guidelines say do not test squamous cell lung cancer patients. So all of this testing is considered investigational. In other words, this is not something where any of the guidelines would recommend that a squamous cell lung patient in this situation undergo molecular testing. Nevertheless, we do feel it is important to notify the patient and the physician upon request or at the time of study closure. Okay, over here? Yes.
Yeah, Dave Rim from Yale, and one of the hats I wear there is I run the division that distributes the tissue for clinical trials, called Clinical Trials Tissue Services. I have a couple of questions related to that. The first is that you require a one-day turnaround. We have a policy of 48-hour turnaround because of the number of clinical trials and the tissue requests we get. Is that going to be an issue?
I'll answer that, yes. So I was actually surprised to see that. So who wants to address this? Do you know what the question is? The submission of the tissue within 1 day of registration?
Yeah. Why do you have 48 hours and not 24 hours?
Well, because we it's a personnel issue and a number of specimens issue. So when you put your request in, you get in line behind the other tissue requests that we have. And we only have 2 people that spend 100% effort processing tissue for clinical trials. And so if it's not, why should we bump your specimen up to the front if there's every other clinical trial also wants the fastest turnaround time possible? We promise, and we have a good track record of completing most specimens in 48 hours, but sometimes we don't get them in 24 hours, and it'd be a shame to see a patient disqualified on that basis.
Yeah, and I can tell you that you are at the top of the list in how fast you do this. I mean, we have tracked a lot of sites, and in some of them it is 5 to 7 days or longer. So I think we need to rethink this.
Well,
my own
let me
opinion.
make a comment.
Well, I think there is, David, an intention. Might not be,
Well, bear in mind that the tissue acquisition is part of the eligibility. So the tissue has to be sent in. So you should probably be coordinating ahead of time with the treating physician. And so when they register, you'll be ready. So
Well,
they
that
should
could be-
alert you.
That's right. The question is, if you just hold the registration until you get the tissue back, then of course we
Yes.
can do it in 1
But
day.
but really,
But if
yes,
the-
you could. But really, you should do that because you shouldn't register the patient until they've confirmed with you that the tissue is available.
Okay, well, I guess that becomes a critical point that maybe
Right.
coordination that maybe should be clarified. The second question related to...
Well, actually, just before we leave this, I think this is going to require a lot of pre-planning then, because although we're talking about it here and you guys say, okay, now I know what we're supposed to do, we're going to have hundreds of sites across the United States. So somehow we're going to have to write this in a way in the protocol where people need to know that they need to pre-plan, as Lucky was saying. Because then if you're already set up and then you register, you've already contacted pathology, you're ready to go. But otherwise, if you start from scratch, I think that's what I was thinking of and you're thinking of, that, yeah. Okay.
Second thing is the rebiopsy. Fred, you estimated that there would be 30% rebiopsy. Will that be a clinical biopsy that goes through the traditional pathology for a diagnosis and thereby sort of obliges us to certain requirements for keeping the tissue? Or will this be 100% research-only biopsy? And have you thought about those implications?
Okay, so there will be two kinds of biopsies, most likely. We think that some patients will require a diagnostic biopsy at the time of recurrence, and that would be a standard of care biopsy. We have actually budgeted 30% of our patients for the cases where the diagnostic specimen is not of adequate quality to provide molecular profiling. And yes, it will go through diagnostic lab as well. So both cases will go through diagnostic lab. It's just that one case will be reimbursed by a different source than the other. And for rebiopsy on protocol at progression, we have budgeted about 20% of our patients. But we would like to encourage the sites to also offer those biopsies to patients so that we can get resistance mechanisms worked out.
So the 30% you mentioned, David, is we're talking here at progression on first-line chemotherapy, right? Because we have insufficient tissue from the early beginning. Yeah.
Now, we haven't talked much about funding, but I think we should also add that that's part of the study budget. So again, we talked about this being a public-private partnership. So the NCI monies that go into cooperative groups already is there, but then there is increased funding, a substantial amount for both SWOG and the other groups, but the individual sites to do all of these other things. So the study will actually pay at a research rate for the biopsy, the processing, et cetera. I don't know if anybody wants to add anything on that.
No, just to reiterate that this is really bringing the biopsies and the profiling, to the entire country. It's very nice.
And then Ray here.
Ray Adesara, Memphis, Tennessee. Valley, the second-line trial, all subgroups compare to docetaxel, so it's probably very important. I'm assuming that docetaxel can't be the second part of the doublet, first line. It's probably very important to emphasize that point because practice across the country includes docetaxel. Will the drug be provided, or will we have to get it because it's sort of standard of care, if you will, the control arm?
It's going to be standard of care, and thanks for making the point about docetaxel. Yes, it is assumed, this is a SWOG study, that many of the sites are actually enrolling on the Frontline study that is ongoing, that is not containing docetaxel. But we will encourage our investigators to use alternative Frontline regimens.
Can I ask how many people use docetaxel frontline for squamous? I think the use has gone down a bit since patents went away.
A lot of my colleagues do it. If you look at community practices, it's a total all over the map kind of thing. Mm-hmm. Then the other question I have then is, if it's going to be standard of care, it takes us at least a week to get approval to deploy a chemotherapy regimen. And so you've got 5 days there. So it's akin to the the 24-hour thing with the biopsies. 5 days may not be enough time.
Right. I agree with that point as well. In other words, because even if somebody gets randomized to standard of care, it can often take over a week to get the authorization, and we're not furnishing the drug. So again, what we don't want to do is have a lot of patients who are ineligible or inavaluable because of things like that. Jim?
Jim Wade, Central Line CCOP. I think there's still open an intergroup trial that is 4 cycles, followed by a maintenance regimen of either placebo or sunitinib that is available for squamous cell patients. So if they were on that study, would that be considered a second line of therapy, making them ineligible for this trial?
Valley, do you want to handle that one?
Yeah, no, we don't consider maintenance as long as it's the same as the frontline therapy. So I'm not aware of the details of the trial that you.
It's a platinum doublet, usually carboplatin paclitaxel, followed by sunitinib, which is an oral VEGF TKI,
Yeah.
among others, or placebo. And so you would have to try to get special permission to break the code if you were saying that sunitinib, which really isn't one of your targets here, was considered as a second line of therapy.
Unfortunately, in that scenario, the sunitinib is considered second-line therapy for study definitions, because if it were part of the frontline regimen, it wouldn't be.
In other words, you're saying because it's a switch
Right.
maintenance. So the issue is we won't even know if the patient got it. That's what Jim was saying, because it was blinded. So you'd have to, that's an ECOG trial?
Yeah.
Yeah. I think that patient would have to be unblinded then.
Yeah, you can, in some of the instances, get permission, but it's hard. The second question would be, in patients who had 3B disease and got treated definitively up front, and then they relapse, have they had, are they then considered refractory to frontline therapy or not?
Yes, it depends whether there was a systemic component to this therapy, but yes, they're potentially eligible.
Well, they're, in giving them either cisplatin etoposide or carboplatin paclitaxel, it would be systemic therapy with concurrent radiation. So when they recur, they'd be considered to be in the second line at that point. Okay.
Paul?
Yeah, Paul Hesketh, Leahy Health.
Yeah.
Just given the complexity of the study, I just want to extend the questions relating to funding. So clearly we talked about the biopsy. So if a biopsy is required, really if it's inadequate at initial assessment to really do the analysis, that would be reimbursed through the study. And if a research biopsy had to be done later on at time of progression, that would be reimbursed. Are there other components of the just the protocol itself? I mean, is there going to be a more ambitious funding mechanism for the sites, other than sort of just specific procedures?
Maybe Nathan Erickson, do you want to talk about the overall scope of the budget? And I mean, we can't probably tell you all the details.
Yeah, but just in a general sense.
Yeah.
Yeah. So this is a NCI-sponsored study, but it has substantial industry support, as was mentioned earlier. On the NCI side, sites will receive the standard payments that have been established within the National Clinical Trials Network system. And those payments will be supplemented with industry support. So in the case of the non-lab sites, which are the most straightforward because they don't receive separate grant money from the NCI, for each accrual to the study, the site would receive the standard $2,250
You're all going to want to sign your patients up for this.
payment, and that payment would be supplemented with industry dollars at a rate of $2,640.
And then that comes through SWOG from the foundation of the NIH, correct?
Yes, that's right. That's right. And SWOG for because this is the study will be open across all the groups. SWOG will pass that money through the other groups to pay those sites as well. For a patient that's screened that doesn't end up going on a study, there's a standard screening payment, which in the new NCTN will be $500. There's also the standard tissue submission payment, which is also established at $500. But that payment is also supplemented with additional industry support, so that sites will receive an additional $479 for that.
And I realize there's still additional questions, but we have 1 minute and 49 seconds left. So I would like Frank DeSanto to talk a little bit about the communications, PR for the study, how we're going to increase awareness and
Sure. The group of partners actually have put together a public affairs committee. We've got representatives from the Foundation for NIH, from Friends of Cancer Research, from all of the industry partners, and from a number of advocacy organizations as well. And we've got a pretty extensive plan for publicizing the study, which includes getting in touch with, and this has already begun, getting in touch with the communications folks at all of the cancer centers out there. And we also have a number of pieces of literature we're developing for patient education, also a patient education video, and we have a launch day publicity plan in place that will include op-eds and a number of major publications, that will include extensive outreach to press, many of whom have actually already covered the Lung Master Protocol. There's already a lot of interest out there.
We're going to be doing, for instance, study-specific videos. One in particular would be on management of toxicities of the anti-PD-L1 sort of immunotherapies, and also the issue about pseudo-progression there. So these will be both for physicians, nurses, CRCs. Roy?
And I think, as Frank mentioned, we're also reaching out to the advocacy groups on our different committees. We have a large number of patient advocates and trying to spread the word about the trial throughout the country.
Are there any further final comments from the presenters or the panel before we close the session? We will be happy to entertain further questions at the reception. Valerie, closing remarks?
I thank everyone for taking all this time to listen to us today, and please go to the website. There are more slides there, and the questions are all welcome.